Good afternoon, everyone. Welcome to ASAP online classes. Today we will be having the session of molecular biology, that is post-transcriptional modification of mRNA. We will be having Dr. 𝐶𝐲𝐽, Professor, Biology, College, 𝐶𝐲𝐽. Dr. 𝐶𝐲𝐽 has graduated from MG University and did her post-graduation in marine biology from Cochin University of Science and Technology. She got her doctoral degree in 2011 from the Department of Marine Biology, Microbiology and Biochemistry, Cochin University of Science and Technology. She has 15 years of teaching experience in UG and PG level and has guided many BSc and MSc projects. She has published many articles in international and national journals, and she has attended many national and international seminar, conferences and workshops, and has convened one national seminar. Now let us begin the session. Over to you, ma'am.
Thank you, Erika. Thank you for the nice intro. Okay. Once again, thank you, ASAP, for giving me yet another chance to present a session. And today we are going to discuss about the topic post-transcriptional modifications of mRNA. So in the last class, we have dealt with what is transcription and how it happens in prokaryotes and eukaryotes. So after transcription, these mRNAs which are formed must go through some modifications to perform particular functions in the cell. And these modifications altogether are known as the processing, or it is rather called as RNA processing. So, what is the processing status in eukaryotes and prokaryotes? In prokaryotes, as we all know, that there is no particular membrane barrier happens transcription and translation. So there is no membrane like nuclear membrane in the case of prokaryotes, which acts as a barrier between the transcription and translation. So that we call it a coupled transcription translation, in which the transcription is as the transcription continues, the translation simultaneously happens. So that's why we call it coupled transcription and translation. So, but in eukaryote, it is not the case. In eukaryote, the primary transcript that is formed, the transcription actually happens in nucleus, and after the transcription, this transcript must be transported to cytoplasm to continue the translation process. So that is the difference between these prokaryote and eukaryote. So in eukaryote, particularly in eukaryote, this primary transcript that is formed must go through some modifications before it is being transported to the cytoplasm. And these modifications, we call it
sorry to interrupt. Hello, ma'am.
Hello.
Ma'am, voice ചെറിയ clarity കുറവുണ്ട്. Microphone ഒന്ന് അടുത്ത് വെക്കാൻ പറ്റുമോ?
Okay. അടുത്തിരിക്കും.
Okay.
Okay. ഇപ്പൊ കേൾക്കാവോ?
ഇപ്പൊ കേൾക്കാം.
Okay. So this is the difference between the prokaryote and eukaryote. Here you can see there is no nuclear membrane barrier. So the mRNA that is being transcribed, it is translated as such or simultaneously. But in the case of eukaryotes, you can see here there is a membrane barrier. This is a nuclear membrane. The transcription happens in the nucleus, and after that mRNA is being transported to the cytoplasm, and then here the translation happens. Then, so as we have already mentioned, this primary transcript, either it can be RNA, tRNA, or mRNA. This is a bulk material, or it has some unusual basis and bulk of material, bulk nucleotide present in this RNA. So it is about 2,000 to 20,000 nucleotide this primary transcript. So that primary transcript that is formed just after the transcription is known as hnRNA or heterogeneous nuclear RNA or pre-RNA. It is called pre-RNA. This pre-RNA must undergo some modification. We call it RNA processing, and to be converted into mature functional RNA. So, what is these processes which include in this RNA processing that we will see? So, modification of RNA, modification of mRNA is not a part of, basically it happens in 3 steps. They are 5' end capping, 3' end polyadenylation, and splicing. These are the 3 mechanisms or 3 basic steps that is happening during RNA processing. So, how all these 3 mechanisms are happening, we will see later. So, what is the significance of these post-transcriptional modifications of RNA? So, the modification of these RNA molecule that actually helps the mRNA to be correctly translated into proteins. So, these modifications are needed for the pre-mRNA to be properly translated into proteins. So, if it is to be mediated to translation, it needs to be processed. Then these post-transcriptional modifications of mRNA actually increases the efficiency of the protein synthesis. So that is the basic significance of these RNA processes. Then this is the process that is happening during this RNA processing. Here we can see this is the DNA, and this is the polymerase enzyme, and that transcribes the mRNA. This is the mRNA. So, during transcription, the whole DNA the whole gene is being transcribed into the mRNA. Not a single nucleotide is being skipped. So, the whole new RNA, which forms the bulk RNA or hnRNA, which consists of both wanted as well as unwanted nucleotides. So, after the completion of this mRNA, they must go through some modification.
Ma'am, voice clarity കുറച്ച് കുറവാണ്. Hello, ma'am. Voice clarity കുറച്ച് കുറവാണ്. Voiceന്റെ clarity കുറച്ച് കുറവാണ്.
അത് ഒന്നും ചെയ്യാൻ പറ്റില്ല. ഞാൻ ഡെസ്ക്ടോപ്പിൽ നിന്നാണ് സംസാരിക്കുന്നത്, മൈക്കൊന്നുമില്ല.
മാം ഒരു കാര്യം ചെയ്യാമോ, മാമിന്റെ വീഡിയോ ഡിസേബിൾ ചെയ്യാമോ? ഓഡിയോ മാത്രം മതി. അപ്പൊ നെറ്റിന്റെ യൂസേജ് കുറച്ച് കുറയും. Okay, ma'am. Continue.
Okay. So after that, it goes through some modifications like cleavage, polyadenylation, and splicing. So the first process of RNA processing is an addition of 5' cap. So what happens during 5' cap addition? So a 5' cap is a residue of guanosine, which is added, methylated at the 7th fluorine ring, which is being added to the first nucleotide of an mRNA. So this occurs just after the initiation of RNA synthesis, or just after about 20 to 30 nucleotide is being synthesized. This capping will be done. So this capping will be done with an unusual 5' to 5' triphosphate linkage. That is between the guanosine as well as the first nucleotide of the mRNA. So this 5' capping actually happens in 3 steps. In the first step, what happens is that a 5' triphosphate at the end of the mRNA, which will be having 3 phosphate groups. And from that, one phosphate group will be cleaved off by an enzyme phosphohydrolase. And in the next step, a guanyl transferase, which transfer the guanyl group or the guanine residue to the first nucleotide. So, to the linkage known as 5' to 5' triphosphate bond. And later, in the step 3, a methyl transferase will transfer a methyl group or add a methyl group to the guanosine. So these are the main steps that are involved in 5' capping. And we will see how this actually happens. So through this diagram, in this diagram, we can see how this nucleotide is being capped. The first nucleotide is being capped with the 5' guanosil, methyl guanosil. So this is what happens. That is, this is the nucleotide, the 5' nucleotide, which is having triphosphate, 3-phosphate attached to the 5'. And the enzyme phosphohydrolase that removes one phosphate from this 5' end. And later, another enzyme known as guanyl transferase, which removes 2 phosphate group from the guanine or guanosil, and then adds that guanine to the 2 phosphate group on the first nucleotide. So forming the guanine 5' cap. Guanine will get attached to the 5' cap. And then in the next step, the methylation of the guanine takes place, and that process is being catalyzed by the enzyme guanyl 7-methyl transferase, in which the methyl group will be added onto the 7th position of the base, guanine base. And after methylation, another methylation will happen. That is on the 2' position of first and second nucleotide on the mRNA, which will be catalyzed by another enzyme known as 2'O methyl transferase. So this is the process of 5' capping, and this is the nucleotide base after the capping, 5' capping. So here we can see this is the methyl group that is being added to the 7th position on the guanine. So, and after the methylation, this guanosil or the 7-methyl guanosil will be attached to the 5'. The 5' carbon of the guanosil is attached to the 5' phosphate group of the first nucleotide. So this is the unusual bondage. Usually in the sugar phosphate backbone in DNA or RNA, either in DNA or RNA, usually this 5' carbon will be bonding with a 3' phosphate group, a 3' OH group. Here it is an unusual bondage in 5' capping, where the 5' of the first, that is the guanosil, will be attached to the 5' of the first nucleotide. So forming the 5' methyl guanosil cap. So what is the significance of this 5' or capping? This cap actually increases the stability of the mRNA. How it increases the stability is that it protects the mRNA from degradation by ribonucleases. So after its function, this mRNA performs its function in cells. This mRNA will be attacked by nucleases, and the attack of these nucleases is being prohibited or inhibited by the 5' cap. And it also enhances transport of mRNA from nucleus to the cytoplasm. It also initiates the translation by positioning the mRNA to the ribosome. And also in translation, there are some proteins are named as cap binding proteins. These cap binding proteins actually recognizes the 5' cap and attaches to it and initiate the translation process. So this is how this 5' cap actually augments the process of translation, and this is how it actually increases the efficiency of translation. So then that is all about 5' capping. Then coming to the poly A chain. So addition of poly A chain. So next RNA processing, the mechanism of RNA processing includes the addition of poly A chain, which is also important as that similar to the 5' capping. So it is the addition of about 50 to 250 adenine residues to the 3' OH of a mRNA. This is what is happening during polymerization. So it actually happens after the transcription. So there are two main steps are included in this polyadenylation. First, the mRNA is cleaved at a particular point known as a cleavage site at the 3' end. So thereby freeing the 3' origin. So later, these adenine residues will be added onto the free 3' origin. So this is what happens during polyadenylation. So this is what is happening during polyadenylation. That is, adenylation is the, as we have already mentioned, adenylation is the addition of polyadenylate residues to the 3' origin of a mature RNA or mRNA. And this particular adenylation happens after these particular termination sequence is being documented. That is known as the AAUAAA sequence. And which you may be remembering, in last class we have discussed about the termination of transcription in eukaryote. We have mentioned about this AAUAA sequence, which is the termination sequence that is being identified by or recognized by the RNA polymerase enzyme. So this particular, when it reaches this particular site or when it is being transcribed in an mRNA, after this AAUAA site, there is 10 to 30 nucleotide downstream from that after this AAUA. That particular portion which is highly conserved, and that is known as the cleavage site or the polymerase site. So here in this diagram, we can see this is the AAUA sequence, and downstream of this AAUA sequence, there is another sequence known as poly-A site. This is the cleavage point where the mRNA is being cleaved. So after that, we can again downstream to that poly-A site, we can see a GU sequence, which is present about 20 to 40 nucleotide downstream of this cleavage site. So this is how this poly-A tail is being added. Then what are the steps involved in poly-A tailing? In poly-A tailing, there is one particular enzyme or factor, a protein that assists in poly-A tailing is cleavage and polyadenylation specificity factor, or CPSF, which actually binds to this cleavage site. So this cleavage site, as we have already mentioned, it is the downstream of, or 20 to 30 nucleotide downstream of this AAUA site, which is very conserved sequence. And this particular cleavage and polyadenylation specificity factor, which binds to this cleavage site or poly-A site and cleaves at that point, and removing the rest of the mRNA from the pre-mRNA transcript. And after this cleavaging, another enzyme known as polyadenylate polymerase, which is a polymerase enzyme which adds adenine residues to the free 3' origin where the cleavage is being occurred. So this polyadenylate polymerase adds adenine residues to this 3' origin. And later, another protein known as polyadenylate binding protein, or PABP, which attaches to this poly-A tail. So it enhances, or it actually protects the poly-A tail, so that no nuclease will attack or be degrading this mRNA. So these are the steps that is involved in polyadenylation. So the main protein that is involved in this polyadenylation is cleavage and polyadenylation specificity factor. So this is the step that is happening during this polyadenylation. Here we can see the DNA that is being transcribed to the mRNA. This is the pre-mRNA. Here we can see the AAUA site, and here after the AAUA site, downstream from that site, we can see 11 to 30 nucleotide downstream from that. We can see the poly-A site or the cleavage site. To this poly-A site or the cleavage site, a factor known as CPSF, or cleavage and polyadenylation specificity factor, which binds, and after the binding, it cleaves off that particular portion of the mRNA, leaving the 3' origin, After that, the polyadenylate polymerase enzyme adds a stretch of adenine residues to the 3' edge, forming a polyadenylate chain. So it may be lengthened about 250 nucleotides, to about 50 to 250 poly A or polyadenosine triphosphate will be added during this polyadenylation process. So this is the process of polyadenylation. So then, what is the significance of this polyadenylation? As in the case of 5' cap, poly A chain also provides protection to this mRNA by the degradation by nucleases. And it also helps in exporting the mRNA from nucleus to the cytoplasm. It increases the stability of mRNA, which acts as a binding site for specific proteins, which are known as the poly A binding proteins. So once these poly A binding proteins is attached to this poly A tail, these nucleases cannot attack or degrade from the 3' end. So these nucleases are exonucleases, ribonucleases are exonucleases, which can either attack from 5' end or can from 3' end. So this 5' cap protects the mRNA by the degradation from nucleases, and the 3' poly A tail will protect the mRNA from nucleases attacked by the nucleases attack. So the length of this poly A tail actually determines the stability of it. So the longer the mRNA, the longer the poly A tail is, the longer the lifespan of it. So that is the significance of poly A tail addition in mRNA. Then coming to the splicing of messenger. So this is the final and the third step or the mechanism of RNA processing, that is the splicing of that mRNA. And splicing, as we know that this heterogeneous RNA or heterogeneous nuclear RNA, which is a bulk RNA, which contains both exons and introns, or exons, which are known as the expressed sequences, which are the segments or the sequences which are destined to be present on the functional mRNA or the mature RNA. And it is these exons will be intervened by another sequences known as introns. They are known as intervening sequence. These sequences in the mRNA are destined to be removed from the pre-mRNA. So that is the removal of these neutrons. That is the intervening sequence from the pre-RNA is actually what is happening during RNA splicing. So we can see here the removal of introns from the primary mRNA is known as RNA splicing. And this is how this RNA splicing happens. And RNA splicing happens once this RNA is being completely synthesized and before it is being transported to the cytoplasm. So this is the exon here. This is the pre-mRNA having the exon 1 and the exon 2 intervened by an intron. So this intron must be removed. So after transcription, it forms the pre-mRNA having the exon 1 and the exon 2 intervened by the intron. And during RNA splicing, this intron will be removed and these two exons will be rejoined together to form the functional or the mature RNA. So this is what happens during RNA splicing. And next we will see what in detail what is the factors which are involved in the splicing and how this splicing happens. So before that, we will have to understand that there are actually different types of introns, and these are based upon how they get spliced. So these introns are categorized on the factor that how it gets spliced up. So on that means there are three. It is being categorized into three. They are group 1, 2, group 2, and group 3. So we see what is the difference between these three kinds of neutrons. The group 1 introns, they are self-splicing, or they doesn't need any protein or support of the protein for splicing nucleus. So no proteins are involved in the splicing mechanism. They are self-splicing, and this self-splicing property is because of the fact that these introns itself access. Introns are actually RNAs. So these RNA itself access enzymes. Then we call it ribosomes. So it accesses ribozymes and will augment or brought about the splicing of the introns from the cells or the brain. And they are found in some nuclear, mitochondrial, and chloroplast genes that code for RNA, mRNAs, and DNAs. And the cell splicing introns were first discovered on rRNA in Tetrahymena thermophila, which is a protozoan cilia, by Thomas Seck and colleagues in 1980. So this is the mechanism of splicing in group 1. Here we can see this yellow color is denotes the introns, and the blue color on either side denotes the exons. And the intron is having a guanine, and the 3' OH of the guanine that reacts with the 3' end of the exon, and it splices it at this point, and so that releasing that end of the intron. And later this guanine will be attached to this end, 5' end of the intron. And in the next step, this 3' OH free end of the exon, which react to this 5' end of the second exon, and it releases that intron from that point, so that intervening intron will be present in the middle of these two exons will be released. And after the release of this intron, these two exons will be rejoined together by RNAs. So this is the mechanism of splicing in group 1 intron. Then coming to the group 2 introns. Group 2 intron is just like group 1 intron. They are also self-splicing, and they are also found in primary transcripts of mitochondria or chloroplast mRNA, fungi and plants and protists. So this is also self-splicing. They doesn't need any proteins to assist in the splicing mechanism. So this is the mechanism of group 2 intron splicing. Here we can see an intron which is intervened by in between these two exons. And the first process is at the 3' end of the intron, there is an adenine, and the 2' hydroxyl group of the adenine will react with the this 3' end of the exon, and it releases or splits that end, and this end, 5' end of the intron, will be attached to this 3' end of the intron, forming a lariat shaped structure. This is what happens during second group 2 mechanism splicing. So the adenine with its 2' OH end that reacts with the this 3' OH of the exon, and it splits the intron from that, and that 5' end will be attached to this adenine, and that lariat forming structure, after the formation of the lariat-like structure, this 3' OH of this exon will react with this 5' end of this exon 2, and resulting in the breakage at this point and releasing that lariat-like structure. And these exon 1 and exon 2 will be joined together by RNA. So this is the process of mechanism in intron 1 and intron 2 splicing. So group 1, group 2 intron splicing. Yeah, next coming to the group 3 introns. Group 3 introns, they are the largest class of introns. They are not self-splicing. They are now self-splicing, and they need some proteins and RNAs to assist in the splicing process. And these complex of RNA and proteins are together known as spliceosomes. So actually these group 3 electrons need spliceosomes to actually brought about the RNA splicing in the in the RNA. So this is what is happening during RNA splicing. So RNA splicing, these introns in pre-mRNA of the mRNA are excised by complex RNA protein complexes, which are known as the spliceosomes. These RNAs that are involved in these spliceosomes are known as snRNAs or small nuclear RNAs. They are known as small nuclear RNAs. And these small nuclear RNAs are associated with some proteins. They are called small nuclear ribonucleoproteins. So when these small snRNAs associated with the proteins, we call it snRNAs or SNRs. It is designated as SNRs, denoted as SNRs. So these SNRs, along with some proteins, actually forms a spliceosome and brought about the splicing process. And these SNRs, there are 5 different kinds of SNRs. They are designated as U1, U2, U4, U5, and U6. So next process we will see how these nerves or spliceosome actually promote the splicing process in RNA. So the functions of snRNAs are that it recognizes the 5' splice site and the branch site, and it brings those sites together so that it can be spliced out. 5' and 3' splice sites are there, and between that you can see a branch site. All these sites are getting together or got together by means of these nerves and catalyzing the process of RNA. So these are the 3 main functions that is being done by the nerves. So this is how this RNA splicing happens. So before we talk about the spliceosome, we have to understand there are 3 main sites that are needed for this RNA splicing. First is known as the 5' splice site, which is denoted by 5' GU. And there is another at the 3' end, which is known as the 3' splice site, which is denoted by AG3. So this is having a sequence of AG, and 5', which is having a sequence of G. And this and another region known as the branch site. This branch site is having an adenine residue on it, and this is where the cleavage happens. So the first factor, that is the U1, SNRP U1, which attaches to the 5' site. So the 5' splice site, it attaches to the 5' splice site, and U2 attaches to this branch site, and later U4 and U6 attaches to this complex, and this U5 actually attaches to the 3' splice site along with the U4 and U6, and this U6 complex with U4 makes the spliceosome transesterase. So this is the process. So here you can see in this diagram, here these are the exons, the red colored or the pink colored of age, and then in the next process, the U1 binds to the U1 SNRP bind to the 5' splice site. Excuse me. Okay, so the U1 binds to the 5' splice site, and the U2 binds to the branch point, which is denoted, which is having an adenine residue. So U2 binds to the branch. And in the next step, U4 and U6 and U5, all these three nerves will attach and form a complex, and that is known as a splice also. So binding of this U1, U2, U4, U6 and U5 actually brings about these splice sites together. Okay, so in the next step, the 5' splice site is being cut. So here, where the U1 is being attached, the 5' splice site is being cut, and that 5' splice site will attach to the branch site, forming a lariat-like structure. So 5' splice site will be released and joins the branch site where the adenine residue is there, and which forms a lariat-like structure. And after the lariat formation, the U1 and U4 will be released from the complex. And after that, the U5 will cut the 3' splice site, and the 3' splice site will be cut. And these so that the intron will be released. So the released intron with this these nerves, all these will be released, and the resulting the exons, these two exons will be joined together by RNA. So this is what is happening in RNA splicing, which are augmented by the spliceosome or SNPs. So that is about post-transcriptional modifications, which include 3 steps. They are 5' capping, polyadenylation, and splicing, RNA splicing. Next, we will go on to alternative, another topic known as alternative splicing. Alternative splicing is also a part of this mRNA processing, where we already know that one gene, earlier we have the idea that one gene actually translated into, one gene is being transferred into RNA. That mRNA transmitted a single copy, but it's not the same case that's happening in reality. Here, pre-mRNA are spliced in several different ways so that a single mRNA can code for multiple copies. So that this is being brought about by the process known as alternative splicing. So the generation of different mature RNAs from a single mRNA, is that occur by varying the splice site, that is 5' and 3' splice site. So the individual splice site can either be activated or skipped. If the splice site is being activated, that exons will be removed. And if the splice site is being skipped, if the splice site, that particular exon is to be skipped, then that will not be detected by the spliceosome, and that will be included into the mature. So that will be a part of the mature. So in that case, these mRNA, from a single mRNA, different varieties of functional mRNA can be produced. And this actually increases the complexity of proteins in vertebrates. So otherwise, these vertebrates, we know that this simpler organism and the evolved organism or the higher organism, the number of genes are almost same. Very much. But the proteins that are produced, now the number of proteins in the simpler organism is much lesser when compared to the higher organism. And this is brought about by the process known as alternative splicing. And this increases the biological complexity of the proteins. So this is what happens during alternative splicing. Here we can see this is the DNA. The DNA is having all these nucleotides, and that is being transcribed into the mRNA, forming the primary RNA transcript. Having all the nucleotides is being transcribed into form the mRNA. This is the pre-mRNA. The pre-mRNA, after splicing, after alternative splicing, we can see here, these are the exons. Exons are numbered as 1, 2, 3, 4, 5, and 6. So the pre-mRNA having all the 6 exons. While in the first set, we can see only 1, 2, 4, 5, 6. So the third exon is missing in the case of first protein. And in the second protein, we can see only 1, 3, 5, 6. That is, the second and the fourth exon is being spliced out. And in the third protein, we can see all the exons is being included. So all these 3 steps of alternative splicing result in 3 different kinds of proteins. So from the single mRNA, pre-mRNA, all these 3 kinds of proteins can be produced. And that is the importance of alternative splicing. So a single gene can actually code for many different kinds of proteins. That is the significance of splicing. So how this is being done is whether a site or splice site is being activated or it is skipped. This activation is done by some proteins known as splicing enhancers. So once the splicing enhancers attaches to the splice site, it means that that particular splice site must be removed. So it is removed. And if it does not attaches, the splice site is being removed, that exon will be removed. It is skipped and the exon will be included in the end. So that is how this alternative splicing help produce different varieties of proteins from a single gene. So thank you. So that is all about mRNA processing in prokaryotes and eukaryotes. So thank you. If you have any questions, you can ask.
Okay, ma'am. Ma'am, നമുക്ക് കുറച്ച് questions കിട്ടിയിട്ടുണ്ട്. ഞാനത് വായിക്കട്ടെ ma'am.
Oh, sure.
How is the initiation of transcription different in prokaryotes and eukaryotes?
Initiation, transcription. Transcription is another topic. Actually, we have mentioned here, we have discussed today about the post-transcriptional modification.
Okay.
It is not about the transcription process. Initiation of transcription in prokaryotes, it is different because in eukaryote we need some initiation factors. Like transcription factors are needed for initiation of transcription in eukaryotic transcription. But while in prokaryotes, it is the RNA polymerase along with the sigma factor does the process. It is actually not today's topic. It was earlier topic that I discussed in the earlier days, the last session. Okay.
And another question is, how does post-transcriptional processing increase the efficiency of protein synthesis?
Efficiency, yeah, definitely efficiency is being increased, as we have already seen that poly-A-chain increases the efficiency by increasing the stability of the mRNA and the transportation of this mRNA from the nucleus to the cytoplasm is done by, or it is augmented by, this poly-A-chain. And same is in the case of this 5' capping. This 5' capping, guanosine capping, 7' guanosine capping actually increases stability of mRNA and thereby promote translation. And it also enhances the efficiency or improves the efficiency of translation.
Okay, ma'am. Then another question is, what can alternative mRNA splicing generate?
So, not clear. What?
What can alternative mRNA splicing generate?
It generates the different kinds of, I don't understand it. It generates Alternative splicing is a process in which a single mRNA can be translated into different many kinds of proteins. By alternative splicing, that is the removal of some exons from the pirion mRNA. So, which exons must be maintained and which exons must be removed, that is being determined by this alternative splicing. So, that result in different kinds of proteins.
Okay, ma'am. Then, ma'am, another question is, can you explain cell splicing property of introns?
Cell splicing, we have already mentioned it in earlier slides. There are group 1 and group 2 introns. Do I need to explain these?
Ma'am,
What is the question?
can you explain cell splicing property of
Process properties.
Property.
This is a group 1 intron splicing here. This guanine residue of this intron will react with this end and releasing that, and this guanine will be added onto the 5' end of this intron. And later this 3' OH will react with this 5' end of this second exon and remove it from that. So releasing the intron, and these two exons will be joined together later. And this is the same case in group 2 introns. Here in this 3' end of this intron, we can see an adenine residue. The adenine, the 2' OH end of this adenine will react with the 3' end of this exon 1, and that makes a cut in between the exon and the intron, so that that particular 5' end of the intron will be released. That 5' end will attach on the adenine, and this 3' OH end of the free exon 1 will react with this 5' end of this second exon, releasing the lariat structure, and later exon 1 and exon 2 will get joined together by means of RNA ligases. So this is RNA cell splicing.
Okay. Then the next question is, what is the mechanism of alternative splicing? Is it also involves spliceosomes?
Yeah, definitely. It involves spliceosomes. The spliceosome detects whether, as I have already mentioned, there are some proteins known as splicing enhancers. So these splicing enhancers actually binds to the splice site to signal the spliceosome that this particular site must be spliced out. If that splicing enhancers won't attach to the exon, that particular exon will be skipped, and that will be added on to this this mRNA, that will be retained in the mRNA and translated into proteins. Definitely, spliceosome is involved in alternative splicing too.
Okay, ma'am. Then another question is, ma'am, why translation of an mRNA can start before transcription of the mRNA is complete in prokaryote but not in eukaryote?
I didn't get.
Why translation of an mRNA can start before transcription of the mRNA is complete in prokaryotes but not in eukaryotes?
Eukaryote. Yeah, you can see in the diagram. We have already, I already mentioned all these things, described all these things in the first lab itself. Here, this is the prokaryote. We know that, you all know that prokaryote is not having a nucleus, a particular nucleus. We cannot call it a nucleus because the nuclear membrane is lacking. So, after the transcription, there is no membrane barrier is there. So the transcription, as the transcription proceeds, the translation also happens simultaneously. These are the ribosomes attached to the transcript. So that is why we call it polysomes in prokaryotes, because one mRNA will be traversed by several ribosomes. But in the case of eukaryote, this transcription happens in the nucleus. So there is a nuclear membrane barrier is between the process of transcription and translation. The translation in eukaryote happens in the cytoplasm. So this mRNA that is produced after transcription must be transported through the nuclear membrane into the cytoplasm. Only then the translation process can be proceeded.
Okay. Ma'am, then one last question is, can alternative splicing increase the biodiversity of proteins?
Yeah, definitely. The diversity of the diversity of proteins is being definitely increased by the alternative splicing. That's why the eukaryotes or the higher organism is having so much different kinds of proteins, and they are all, say all, but most of these are related. They are very much related. But because these are the precursor of these proteins are single mRNA, and that is brought about by the splicing process. So the diversity is being increased by this alternative splicing.
Okay, ma'am. There is another question. What are the resultants of abnormal variation in splicing?
What are the
resultants of abnormal variation in splicing?
Resulting this mutation. Any mutation, if it happens in the DNA, it will be definitely happens in mRNA. So if any mutation happens in DNA, that will be transcribed to the mRNA, and that will be translated to the or present in the proteins. So definitely there will be.
Then another question is, how the poly A tail and 5 cap function in translation?
I have not answered the previous question.
Hello,
it's
ma'am.
Not completed.
Okay, ma'am. Then continue, ma'am.
So the mutations happening in the DNA can produce improper proteins and not formed or regular, not functional or non-functional proteins, and which definitely affects the cell process or cells regular processes. So or regular biochemical processes. So that is how these mutations and improper arrangement of these nucleotides incorporated in this mRNA or the DNA will affect the transcription as well as the proteins that is produced by translation. Then the next question.
How the poly A tail and 5 cap function in translation?
Poly A tail translation, because it increases the process of translation because it actually helps in transporting the mRNA to the cytoplasm. Then only it gets translated. And in the case of 5' cap, there are some proteins known as cap binding proteins, which helps in translation. So these cap binding proteins first attaches onto this 5' cap, and then after that this translation process can continue. So that's why that's how this 5' capping in the poly A tail helps the translation process.
Okay, ma'am. ഒരുവിധം relevant questions ഒക്കെ ma'am ഞാൻ ചോദിച്ചിട്ടുണ്ട്. So I think it is time for us to wind up the session. And thank you, ma'am, for spending your valuable time with us. And thank you, attendees.